All right, hi guys. So give me like 2 seconds to set up. Today we're gonna do a little bit more biology because it's been a while since we've done that. Last few times we've been doing chemistry, so we'll be doing a little bit of chemistry in the biology. We'll see about it. Just give me a minute or so, and I'm just setting up some stuff. All right, so I'm gonna share screen on the iPad. All right, cool. So I'm gonna go over here. Yeah. Start out with this. All right, so I think the way I have it planned today is that we're gonna work mostly on the cell transport aspect of biology because um, Larry's asking a question that I was also planning to touch on anyway. Is biology just memorization of proteins and ecology and cells and anatomy? Um, so there's going to be more to biology than just those four topics. Of course, there's a lot to biology, but uh, what I observed, at least from my experiences with biology, is that it is quite a bit more memorization based, and there's a reason to that. It's because with with sciences like physics and chemistry, uh, the underlying foundation is principles. So like kinetics for chemistry, equilibrium, or if you look at physics, for mechanics, there's kinematics, energy, momentum. These are different models that describe systems, and you could take these models and look at any different kind of mechanical system for physics, and then describe that system using these principles. Same thing with chemistry. You can take equilibrium, you can take kinetics, you can take uh, other elements, solutions, and describe chemical systems with them. With biology, you're looking at a system and describing it. You're like, it's like the difference between explaining and predicting. Chemistry and physics have principles that you can use to apply to new situations. Whereas usually with biology, say like the human anatomy, you don't really apply that to another thing. You just know the human anatomy and know more about it, and with biology, there is, of course, some principles that you can then apply to new situations. A lot more of biology, though, is learning how to explain something, like how to explain anatomy or explain what cells are like. Cells aren't principles where you can look at a different kind of some weird new cell and then have a new principle. Yes, there are different kinds of cells, but then you would learn you'll learn more about that specific cell, in which case you're getting back to the memorization part. So anyway, biology is more explaining what's already there, whereas physics and chemistry, there's more of like a translational purpose to it. This is one of the topics in biology where it's like foundational to other things in biology. So you can use cell transport and the ideas of cell transport to explain other systems. So this is probably going to be it was one of the only topics in biology that I could find that I feel like is worth our conversation, our discussion, because if I were to sit here and teach you guys anatomy, I'd just be showing you a diagram and you'd stare at it for an hour and try to learn as much as you can. And, that's not really a discussion, is it? So with biology, there is, of course, strategy to how to study and all of that. But when you ask, is it memorization? Yes and no, but yes. Anyway, um, let's start up on this. So cell transport. The first place we need to start is with these down here, labels there. So that's a phospholipid. A few weeks ago, we actually did talk about biomolecules. So I'm wondering, does anyone remember what a phospholipid was about? Or perhaps first, and it's in the name phospholipid. What category of molecules is it? Sorry there.
It's a lipid.
It's
My
a
mic
lipid.
wasn't working.
All right, no problem. Yeah, it's a lipid. So a lipid is a very unique kind of class of biomolecules because it's basically anything that's water insoluble. So nucleic acids are water soluble, carbohydrates are water soluble, amino acids are definitely water soluble, although they have parts that are not. Amino acids are unusual, but lipids are probably more unusual in that it's a very loose category, pretty much anything that's water repellent. For example, a steroid, I'm just going to draw a generic set of odd looking hexagons. Usually steroids are carbons attached in like 3 to 4 hexagons or pentagons of some kind with, of course, different attachments and all of that. They are also water soluble because they're mostly carbon and hydrogen, which are not very polarized. Oh, I'm saying a lot of different things that might not make too much sense. Aren't steroids bad for you? So there are synthetic steroids, which of course, if you consume, then those are drug-like. But steroids also include things like cholesterol, which is very necessary for your body. Of course, not in large amounts, in a balanced amount. There are things like hormones, many different kinds of hormones, like estrogen, which develop certain kinds of maturity factors, things like that. Steroids, many of them are in your bodies right now, and they're serving very important purposes in cell communication, different other things, cell membrane fluidity as well. But steroids, if they're synthetic, they're probably bad for you. You wouldn't want to eat a steroid because that's probably not a good steroid for you. But other molecules that are classified as steroids would be good for you, and are very necessary for your function. Anyway, a steroid is a lot of hexagons, which of course would look very different compared to what you see right here, phospholipids, which look like little walkie things or something. Two very different kinds of molecules, but they're both considered lipids because water soluble. That's like the main overarching idea that goes with lipids. So this particular one's called a phospholipid, and the name should make sense because we have a phosphate group in there and there. This one's probably the most simplified diagram on the right, but that's the phosphate group. And attached to the phosphate group let me zoom up on this one. Give me a sec.
2 hydrocarbon chains.
Shoot, it's like, yeah, 2 hydrocarbon chains. You're absolutely right. There's also something else to it. Oh, there we are. Okay. Come on. Give me 1 second. All right. So here are the 2 hydrocarbon. Oh my goodness. All right. The 2 hydrocarbon chains that Angelina referred to, yeah, those are there. And there's also this one thing right here. In this diagram, it's a little unclear, but it's yeah.
Wait, it's a type of linkage, isn't it?
It
Phosphodiester linkage, I think. I'm not sure. I know,
maybe
I just remember reading
I'm
it.
not.
So
Yeah, it sounds familiar. What you're saying, the phosphodiester linkage. I think that's something else. I'm not 100% sure. It might be, but what I was gonna say is that that's a glycerol. So a glycerol is kind of like it's kind of shaped like this. And then these are carbons, and there's hydrogens and stuff attached. Essentially, it's like a three-pronged thing. So if I were to break this molecule down, what I'd see is carbon chain, carbon chain. Attached here, this one is actually going to bend backwards. And there's going to be this phosphate group. Another O as well. So that's how this thing is attached. It's the glycerol that holds these three parts together, the phosphate and the two carbon chains. All right, so that's like the molecular structure. And here's another diagram of it. The simplified diagram, I think, is most clear in emphasizing what the different parts are serving. So we have this thing, this thing saying hydrophilic head and hydrophobic tail. We did mention this before. Does anyone remember what hydrophilic, hydrophobic mean?
Attracted to or repelling from water.
All right, so which one's repelling?
Hydrophobic.
All right, so that one is repelling. Yeah, and then the next one, this one was, what'd you say again?
Attracted to water.
Attracted. Yeah. Attracted. Okay, so hydrophilic head is attracted to water. And the reason for that is because a phosphate group, I think we probably did work with a phosphate in one of the last few classes. Phosphate group is a negatively charged ion. It's PO43 minus, so I need two more O's around there. So PO43 minus, that's negatively charged. And because it's negatively charged, it's going to be attracted to water. When things are not charged or not very polar, then they're not attracted to water. Whereas things that are charged or very polar, they would be attracted to water. Now, why is that? So I'm going to just draw a simplified PO43 minus. And I'm going to draw water. So these are the hydrogens.
You drew, oh, never mind.
Yeah. I'm not going to draw the H's that close, because then it looks like OH. But these are hydrogens, and that's an oxygen. This arrow, this arrow is directing polarity. So what that means is that with water, you're going to have a partial positive at the hydrogen and partial negative at the oxygen.
You drew O. Is that meant to?
Yeah, no, I'm just drawing little circles for the atoms.
Oh, never mind. Sorry.
Yeah, no problem. So what I mean by partial negative, partial positive is charge, because oxygen is electronegative, which means that it's attracted to electrons. And there's a bunch of reasons for it. It's got more protons, and its outer electron shells are farther away, so they're less attracted, blah, blah, blah. So that's why oxygen is more electronegative. It's going to be more attracted to electrons. And hydrogen is going to be less attractive because it doesn't have as much of those things. This little bubble I'm drawing around here is a theoretical electron density around this molecule, because all atoms have electrons, right? Atoms are protons and electrons. When you form bonds between different atoms, the electron density kind of encapsulates the entire molecule, but the electron density can be shifted somewhat. So it can kind of, for just as an example, this would be shifted towards the oxygen. This red electron density would be shifted towards the oxygen. You can also imagine electron density shifted toward the hydrogens. The red one is going to be very roughly, very approximately the kind of electron density we're going to be looking at here. Because oxygen is more electronegative, that electron density is going to be closer to the oxygen on average. So that's why we have a polarity. All right, yeah, lots of chat from Larry. What did hydrophilic and hydrophobic? Okay, hydrophilic is water attracting or water loving, if you want to characterize molecules. I don't like characterizing molecules, but attracted to water, basically. And what is hydrophobic? That is repelling water. So they're kind of opposite. I think you meant to write PO43 minus there. PO43 minus is phosphate. It's like an ion. So if you think about like hydroxide, that's an ion, or NH4 plus ammonium, that's an ion, things like that. Anyway, so water has a polarity. Essentially, one side is negative, one side is positive. And now it's not complete separation of charge. So let's say I had a Na and a Cl. This would be full separation of charge. Cl would have one more electron than it should, and Na would have one less electron than it should. So that's a complete separation of charge. Whereas with water, it's going to be a partial separation of charge. So a little bit of a shift in that electron density. All right, so far so good. All right, so what happens with this phosphate is that since it's negatively charged, remind me again, are negative, is it like charges attracted or opposite charges attracted?
Opposite.
Opposite, right. So positive attracted to negative, negative attracted to positive. So, shoot. Anyway, so what that means is with PO43 minus, it's negatively charged. So it's going to be attracted to positive things, such as those hydrogens on that water molecule. So I'm going to once again draw a little water. I'll use blue to represent PO43 minus. There's going to be an attraction here. A little bit of an attraction. Other water molecules as well. Oh, shoot. Other water molecules also attracted. So on and so forth, until there are water molecules surrounding this phosphate group. Attractions means that that there's, well, positive negative attraction means that energy is lowered. And to explain why that is, I have to go into a bit of physics with how you have higher potential energy when attracted charges are separated, and when they're brought closer, there's lower energy. But essentially, this is a more favorable state, right? If two things are attracted and you hold them far apart, that's not as favorable as when they're together, right? That's why magnets and stuff, they're attracted to each other. So when phosphate is in water molecules will orient themselves in this manner. The red is water and the blue is phosphate, so other way around. There. Anyway, so with phosphate, there's this attraction in water. And since these attractions are favorable, phosphate's gonna be able to dissolve in water very easily. And this is where the idea of hydrophilic, water-loving, comes into play. Water and phosphate ions are attracted to each other. So phosphate is hydrophilic, water-loving. All right, so going back up, then there's the carbon chains. So carbon, if we think about that electronegativity thing we were mentioning earlier, where oxygen is more attracted to electrons, hydrogen is less attracted to electrons. With the hydrocarbon chains, we basically have a lot of carbons and a lot of hydrogens attached to each other. Dot, 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 so on and so forth. So between carbon and hydrogen, there's a reason to this, but they have similar electronegativities. So there could be pretty much equally attracted to electron densities. So there's going to be an even electron density around this hydrocarbon chain, approximately. And between carbon and carbon, of course, they're the same atom. That's going to be same electronegativity between those two. So since there's no separation of charge, there's no partial positive, partial negative like we see with water down here. There's no partial negative, partial positive. There's going to be no attraction to water with hydrocarbons. So that's why this carbon chain right here is considered hydrophobic. It's not attracted to water. It's actually repelled because water molecules are going to be more attracted to themselves than to hydrocarbons. So that kind of causes an indirect repulsion effect. That's a little more complicated, something else for later. Hydrocarbons repel water, yes. But when you say hydrocarbons repel water, that's usually an effect that you see when you have a lot of hydrocarbons. Like if I had one hydrocarbon and I put it in a giant tub of water, just one single molecule in water, Well, it's got nothing to do except kind of stay inside the water. There's not much else to do. Um, so yes, it will remain in water. But if you have like a layer of oil, which is a hydrocarbon, and a layer of water, well, we know oil and water don't mix. That's because water molecules are more attracted to themselves than they are to any oil molecules or any hydrocarbons. So then the hydrocarbons kind of get pushed out of the water, and then they've got no choice but to remain in
And then
this
they
position.
properly named this hydrophobic reactions.
Yeah, exactly. So that's why we have these hydrocarbons that repel water because they're the one thing that's not attracted, whereas everything else is attracted to water. So then they're automatically kind of pushed out in a sense. All right, that's a big tangent, and we went back to chemistry, but back to biology parts. So we have this phospholipid, and we have hydrophilic and hydrophobic in the same molecule. There's a name to that. It's I hope I'm not remembering the wrong name. I think it's called amphipathic.
Amphipathic.
Amphipathic. All right, I got that right. Woo. Anyway, thank you, Angelina. So when we have an amphipathic when we have a molecule that's both attracted and repelled by water, like phospholipids, uh, some cool phenomena can occur. Larry, I'm not answering that. Anyway, phospholipid, what happens, and this is completely natural, right? If you throw a bunch of phospholipids in water, this whole structure forms. And this happens by itself. Scientists have indeed thrown phospholipids in water, and these structures have formed automatically by themselves because it's just favorable. What happens is the phospholipids line up like this and like that. And this goes on. And why does this happen? Well, we can think about the environment of this structure being water. So we've got little water things. I'm just gonna draw them as circles. These are what the circles are water molecules. A lot of them, basically. So does this structure form because the heads like water and the hydrocarbons are repelling water? Right on the point, Larry. So water is attracted to the heads because remember the heads are phosphates. They're negatively charged on average. They're not always, but that's a more complicated story. But they're going to be attracted to water because of those electronegative, negative charge attracted to positive charge kind of thing. So water molecules will indeed be oriented like this with the hydrogens pointing towards negative charge. So there are these attractions, but with the hydrocarbon tails, there's going to be no attraction. So if you imagine there being a phospholipid like that, well, this phosphate now has zero attractions with water, or very few attractions with water anyway, but the hydrocarbons also don't have an attraction with water. So this is simply not favorable energetically, right? You're not optimizing this system to have the lowest energy and to satisfy all the different attractions. So that's why any phospholipid molecule that's oriented like this is just naturally gonna flip around. Because once they flip around, they're not gonna flip backwards as easily because this attraction exists, right? Anyway, what we're left with is in this region right here, this is kind of where, this is like the dead zone. There's no water in this area. And also no charged particles that can really exist here. Because think about it, if you're charged, you have no attractions here. But if you're to shift out, you'd have so many attractions with water, not energetically favorable. So therefore, charged molecules just kind of don't exist here. And you can imagine this structure having many, many, many molecules. So that can curve, and then eventually can curve into a little circle. So just think about like the little phospholipids forming up the surface of this object. And that's kind of the basis for a cell membrane. So Larry did mention this in the beginning to the private chat to me, that this is a cell membrane. The phospholipids are the backbone for cell membranes. All right, that's just introductory. No, I'm joking. Anyway, what happens if you, oops, what happens if you mix salt and oil and then water? Will the salt break up the phospholipids and they will mix? All right, so what you're suggesting is like the phospholipid molecules break apart because of the salt? Because to break apart a molecule, you're going to need, oh, break apart the membrane. Mm, no, I don't think that would actually happen. I think the salt does like water, yes. The membrane will stay together though, because basically the salt molecules, the charged particles will just be ejected from the membrane. Uh, None of the molecules will really be broken apart. Yeah. Okay. Anyway, this is how a membrane forms basically. Phospholipids put in water will naturally form this membrane because energetically, this is the only thing that really makes sense. All right. And you'd be surprised, like if you're looking at AP Bio or you're thinking about doing AP Bio, um, many times the question is going to be like, which of the following is possible in water? And then you're going to have a diagram like this, and you have a diagram like this, and a diagram like this. I've seen an AP question like that before. And like now you guys can solve it. No problem. Anyway, AP aside, AP does some stupid ideas sometimes. Anyway, that's out of the way. And now we'll go up to these other diagrams. So first, how should I do this? All right, what I'm gonna do is I'm gonna erase all this. All right, that's good enough. All right, yeah, and I'll go back up. Okay, so um, with passive, what's up? The first one, the first choice. I'm not sure what you mean there. Anyway, passive and active transport. This is going to be utilizing the properties of the phospholipid bilayer. So diffusion, shown right here. Diffusion's where molecules just kind of travel across the phospholipid membrane. And now we've talked about charged particles. They're not going to be able to pass through the phospholipid membrane because it doesn't make energy sense, right? Uh, so what molecules would be able to pass through? Well, one example of a molecule that can pass through is water. And water, we did talk about how it's polar, therefore it's got some kind of charge, therefore it shouldn't really be able to mingle with hydrocarbons. The thing is, water is a very small molecule, and its charge disposition is a very slight uh, charge disparity. It's only a little bit of a difference between water and oxygen because it's still a regular bond. Hydrogen's not devoid of um, of electrons, so it's still not polar enough and also very small. So therefore, it's actually still able to pass through the membrane. It just passes through at a very slow rate. Another better example of a molecule that can pass through is, for example, let's say methane. Now, methane is a gas, so it probably shouldn't be inside your cells. And if you inhale a lot of methane, that's not very good for you. But for the sake of argument, let's imagine you have a methane molecule that is in the process of passing through your cell membrane. This could actually work because methane in the water environment is not going to be very attractive. But passing through the hydrocarbons, it's it doesn't gain new attractions, but it's not repelled by the hydrocarbons either because it's not attracted to the water environment. It doesn't prefer either environment. So that's why it's actually able to move through the phospholipid bilayer, the membrane. Other examples of molecules would be, say, this molecule right here. I'm just drawing a random molecule here. My point is that with small molecules and also molecules that are not particularly charged, then they're going to be able to pass through the phospholipid bilayer. Now, if your molecule is very big or if it's charged, you're going to have problems. And you can imagine it, right? If you have a really big molecule, how's that going to fit through the phospholipid bilayer, even if it's not charged, right? It's too big. It's like clunky, right? But if your molecule is charged, you're repelled because you're attracted to water. So if you're small and you're not too charged, you're going to be able to pass through the phospholipid bilayer. Does active transport require enzymes? We're going to get to active transport in a little bit, but no, it doesn't always require enzymes. We're going to talk about that.
I think like
Anyway,
carrier proteins or something, right?
carrier proteins, yeah. No, carrier proteins are also not really.
Transport proteins, something like that.
No, those are both passive transport. We'll get to it. Anyway.
Oh.
So starting with just regular diffusion, okay. We see something said here, high concentration to low concentration. That's going to be very necessary for diffusion. I'm going to go down a little bit. And I'll draw, that's going to be my phospholipid bilayer. Not as detailed, but it will serve the purpose. And let's say I have a bunch of small uncharged molecules, okay? Small uncharged molecules. And these molecules are just going about, usually molecules in water, they're going to be moving around a lot. Generally, molecules in gas or water or any kind of environment, they've got kinetic energy and they're going in all different kinds of places, bouncing off each other, bouncing off water molecules, so on and so forth. Eventually, some molecules are going to bounce over. And then this molecule might bounce over, so on and so forth. Random motion will inevitably have some molecules crossing this membrane. Now, if your molecule is charged or large, it won't be able to cross, but since the membrane is not a barrier for these small uncharged molecules, they're going to be able to cross no problem. Now, of course, this molecule might bounce back, but what you might notice is that when you have less molecules on this side, less, and if you have more on another side, then it's going to be more likely for molecules on the more side to bounce over to the less than it is for the less to bounce over to the more. So far, so good? All right. So now I have even number of molecules on both sides. So now it's going to be an equal chance of molecules bouncing over. This is what's called equilibrium, which we've talked about. This is, of course, a very simplified definition of equilibrium. There are more stuff going on in reality, but if there's equal number of molecules on both sides and it's the same environment, same temperature, whatnot, then it will be an equilibrium. Of course, this is a dynamic equilibrium because we generally only really consider dynamic equilibriums. Molecules are always moving, so you're never going to have anything really static, but it's an equilibrium nonetheless. Now, if I have more molecules on this side, now this is no longer less. This side becomes less, this side becomes more. The property acts the other way. Molecules generally shifting this way. That's literally all there is to diffusion. So if I go back up, diffusion is just natural bouncing around of molecules. So what we see right here, essentially. Molecules just randomly bouncing around such that if there's more molecules on one side, eventually over time, the density of molecules on that more side will decrease and the other side will increase as molecules just generally shift to balance both sides. All right, that's pretty much all there is to diffusion, and people call it simple diffusion for a reason. It's quite a simple concept. Now, what about charged molecules or large molecules? Let's say on this side, let's say I have a bunch of these green triangles, as the diagram depicts. So we have more on one side, less on the other. They're not going to be able to pass through the membrane, though. So that's why we have these things called channel proteins. So Angelina, you brought this up, channel proteins. They're actually going to be part of passive transport, not active transport, because here the purpose that they serve is essentially to block out the phospholipids. So there's going to be this channel right here. I can't really zoom up because then my thing will glitch out. But hopefully you guys can kind of imagine there's that little channel poking through the membrane, and that channel is kind of just filled with water. It's like a hole in your membrane. Through that, you'll be able to have charged molecules passing through. Now, of course, a protein channel, such as this one, will have different regulators so that only a specific kind of charged molecule can pass through. But it will still allow that molecule to freely pass through with no problem at all. So that's kind of the same as diffusion, where there's more on one side, so therefore over time, just naturally, you'll have more on the other side, shifting so that you would have equal numbers. But this time you're just bypassing the phospholipid membrane. So now large and charged molecules have their own way of passing through the membrane.
But essentially, it works kind of just like diffusion, right?
Yeah, exactly.
Just goes through something instead.
Yeah. And I did briefly mention that this is a protein. A protein serves this function. Protein serves many functions, but this is going to be a protein. So Larry, you mentioned enzymes. Enzymes are not going to be involved with cell transport because enzymes are specific proteins that break down molecules. Here, you're not really breaking down molecules. You're just sending them one side or another.
Wait, do they have to break down molecules per se?
No, they don't.
They're
Proteins don't
just
have to break
speeding,
down.
right?
Yeah, they're
Or
just
enzymes, they're just speeding up reactions, right?
Oh, enzymes. Yeah, sorry, you're talking about enzymes. Yeah, enzymes can just speed up reactions, but usually, since if we're talking about catabolic reactions, which generally there are more catabolic reactions that biologists study, or at least at this level that biologists study. That's why I said breakdown instinctively. But yeah, you could have reactions, enzymes that catalyze building reactions. Those are anabolic reactions. Enzymes just really catalyze reactions. Yeah, you're right. I shouldn't have said breakdown, although they do break down quite often. But yeah, thanks for that, Angelina. Anyway, here we're not going to be talking about enzymes. We are talking about proteins, and enzymes are a kind of protein. They're proteins that catalyze reactions, that speed up reactions. Here, these proteins are not speeding up a reaction per se. Now, you could say that the transport of a molecule demonstrates one state and another state, and therefore it could be considered a reaction. Most times you wouldn't really consider it like that, though. Anyways, kind of a dilute definition of reaction. Regardless, this protein is serving the function of transporting a molecule, or at least allowing a molecule to pass through the membrane. And if I were to draw like an aerial view of this system of phospholipids, these are going to be all the heads. And then a protein is going to be like this massive thing surrounded by a bunch of these phospholipids. And then there's going to be a big hole in the middle where molecules can pass through. This is just kind of like an aerial view of that. You can imagine it like that. Anyway, taking that away. There's also going to be this thing called protein channels. That's what we have right here. Right here. That's a protein channel. Sorry, not protein channel, my bad. That's a protein carrier, so a carrier protein. Basically, a molecule enters the carrier protein. The carrier protein changes shape and then lets the molecule through. And then changes shape again to receive another molecule and then deposit it. It's also going to be considered a form of passive transport because it happens without energy. It happens based on more molecules being here and less molecules being here, but it does this shape changing thing. So you can consider it a little more regulated than just a protein channel. Carrier proteins by the name are also proteins, but they just behave a little bit differently from channel proteins. All right, any questions so far? That's good. As you guys can see, that this image is from Shutterstock. I erased it at the beginning of today, but that's revealed again.
Oh, for carrier proteins, how exactly do they change shape, or is it? I mean,
That would
different.
depend on the carrier protein because proteins there are many different shapes, but you can here let me draw a little diagram.
Like,
I don't
how
know
would
if
it
this
work,
will.
and what does it depend on? I guess.
What does it depend on? Well, because proteins are not really just like little slabs that go like this. They have very complex structures to them and shapes and things like that. So when a protein changes shape, that's like one part of the molecule like rotating around, and then now it's attracted to this other part of the protein, so then snaps together, which then causes this part to rotate. Protein shape changing is, or protein folding, or anything like that, where the protein like 3D arranges itself in a different way, are very complex mechanisms, and that's why many university researchers and things like that, they study protein folding or protein shape modifications because it's a very complicated process. Um, but what happens here is the protein changes shape because it receives this specific molecule. Let's say this is some kind of ion. Let's say Ca. Oh wow, Ca2 plus, for example. The Ca2 plus would then have a specific place to bind onto the protein. There's going to be a specific place on the protein that's attracted to the Ca2 plus. It attracts to this place, and that attraction is what causes some part of the protein to then twist or change shape or fall apart or something like that. And then the Ca2 plus would then move in because of the protein also changing shape. And then through many different shape changes, the protein is now going to be a different shape, but the Ca2 plus will be on the other side to be let through. It's a very complex process, and each protein is different in that specific regard. Um, so maybe not the most satisfactory answer, I'm sorry, but that's pretty much all we know.
No, it's good enough, I think.
All right. Cool. Any other questions? Because this is pretty much all of passive transport and
Oh,
a little bit.
for active transport, are they pumps?
Yeah.
Is that the thing? That was
Pumps,
the.
yes, pumps. So yeah, protein pumps. So here we have carriers. Oh, the handwriting is bad. Anyway, channels. And then with uh, simple diffusion, it's just nothing. You just go through the membrane. Pumps are over here. So yeah, with active transport, there are pumps. Now, what's the difference between active and passive transport? Well, there's this thing right here, ATP, which is, someone tell me what is ATP for?
Energy.
Energy, yeah. Larry, Angelina, pretty good. Energy. ATP is an energy molecule. It stores energy. Breaking apart releases energy. So ATP is a way to transport energy very easily. The molecule can go around and basically is just portable energy. Here, an ATP connects with this particular pump and provides energy to this pump, and that causes the pump to change shape. And this particular change shape is different from, say, a carrier, because what it does is it takes molecules from a low concentration area and puts it to a high concentration area. All right, so that's where this diagram is wrong. So I want you guys to not really look at this diagram because here it looks like the purple is high concentration going to low. That's passive, not active, and this diagram is kind of faulty for that. Uh, if this purple side were of low concentration, and then you're taking molecules from the low concentration side to the high concentration side, that would be active transport. So if I were to fix this diagram, I just draw a bunch more of these purple little circle things. So this now high concentration side. If you're taking molecules from low to high, that's not favorable, right? Because high will generally want to flow down to low, or just from random collisions, it's more likely for molecules on a high concentration side to then over time shift to a low concentration side. That's not what happens with active transport because there's these pumps actively pushing for molecules from low concentration to high concentration. And that takes energy because it's not favorable. So that's where ATP comes in, provides that energy for this particular transport.
Is that always the case?
Always the case, yeah. You will always need ATP or some other form of energy. GTP.
Oh, I mean from going
Yeah.
from low concentration to high concentration,
Pretty much. Pretty much all the time. That's what's going to be
or
happening.
at least most of the case.
The vast majority. I can't think of a case that's not that particular case. If it's from high to low, it will be some kind of passive transport, maybe a carrier or a channel, but you probably wouldn't be using a pump necessarily. Because carriers and pumps are actually quite similar, as you guys can tell. They both change shape to move molecules. It's just that one requires energy, one does not. So a pump by definition would be one that requires energy, therefore moving molecules from high to low. Sorry, low to high. Pump moves from low to high. All right, any questions? Cool. So final thing right here. This is just like a little note. Also going to be part of active transport. So this is active. Phagocytosis and pinocytosis, these words right here. Phagocytosis is essentially consuming like bacteria or some other kind of living organism. Our cells do that sometimes. They consume bacteria. Or if you think of like an amoeba or some kind of single-celled organism, this is how a single-celled organism would be able to eat. So if you go to a pond and collect some pond water, you're going to have single-celled organisms in there, and they're going to be eating other organisms, other single-celled organisms. And that's going to be through phagocytosis. This diagram shows it pretty well. The plasma membrane kind of curves around, kind of curves around the thing being eaten, and it forms these pseudopodiones or pseudopodia. That's just a large word for these little finger things. The finger things connect around, they connect with each other and they form this little bubble. This particular cell is now eaten by the larger cell, and that's a process called phagocytosis. Moving the cell membrane in this irregular shape would, of course, take energy. So that's why this is still active transport. Pinocytosis is where the membrane kind of curves in on itself, and this time it's to receive molecules. Usually pinocytosis is for receiving water, along with any other kinds of particles that might be in the water. If this water is particularly salty, there will be some salt particles that pinocytosis will be picking up. So curving in on itself, picking up water and whatever's in the water, and forming this little vesicle around it. And it's also another way, as you can think of it like drinking in a sense. This is phagocytosis is eating, pinocytosis is drinking. Don't say that on a biology test, but that's one way you can kind of imagine it. These both will require energy because they're both bending the cell membrane in odd ways. Exocytosis which I didn't bother getting a diagram for because it's actually very simple, is very similar to endocytosis because it's kind of the opposite of endocytosis. Just imagine pinocytosis happening, but the opposite way. Essentially, you have a vesicle. The vesicle comes along and it fuses with the plasma membrane because vesicles are kind of just like mini membranes. They're also phospholipids. So the phospholipids of this membrane can easily just kind of attach with these phospholipids, and then they form one thing, and then they form a little cavity, open up, and then they kind of shoot out whatever the vesicle is holding, and then form the membrane once again. That is exocytosis. Exo meaning you're putting out stuff. You might put out waste, for example. That is something our cells do very regularly. They use exocytosis to put out waste. They might also put out proteins or hormones or communication molecules, neurotransmitters. There's a whole variety of things that exocytosis is used for. Same with endocytosis. It's eating bacteria to digest them and break them down. It's
Wait,
consuming
so
fluid.
is endocytosis pinocytosis?
No,
So then,
pinocytosis
oh,
is endo because endocytosis is eating or drinking or
yeah,
it's
sorry, that's what I meant. Is pinocytosis endocytosis?
yes, pinocytosis is one form of endocytosis. Same with phagocytosis, it's one form of endocytosis. Exocytosis is just putting out, so it's separate. All right, and that's my spiel. Any questions on passive, active, endocytosis, exocytosis?
Yeah. Oh, why
Or
would
just
they be active transport?
why would they be active transport? Because this is not something that, because when something is not active transport, or when something is not requiring energy, because active just means requires energy. If something is not requiring energy, it has to be something favorable. Now, sticking your cell membrane out like this is not something that happens naturally. It's not energetically favorable for you to stretch your cell membrane, just like how a rubber band doesn't just exist stretched at all times. You put it down on the table and don't touch it, the rubber band's going to be in a relaxed form. It's not going to automatically stretch itself because that doesn't make energetic sense. You have to input energy by your hands or something to stretch that rubber band or to bend that cell membrane. So that's why that's where the energy is needed.
What about the parts with like pumps and transport?
Oh, with pumps, because you're pumping from usually every time, pretty much, from low concentration to high concentration. And remember, from low to high is not a favorable transmission. It's like
Wait,
it's
so is that
not,
just one type of active transport?
it's just one type of active
Oh,
transport.
okay, that makes sense.
It's generally just called active transport. So there's that. Um, There's actually one other kind of active transport called secondary active transport that I don't talk about today because it's a little more complicated. It's where you have one side being favorable and another side not being favorable. It's like basically passive and active happening at the same time. It's kind of more complicated. Uh, and also probably something that if you're interested in, you should do some further reading on. For today's time and purposes, not something we're going to be able to go over. Anyway, any final questions about this? Because there's one last thing I want to do, but I want to make sure this all makes some sense before we move on. All right. So let me bring it up. All right. I am going to share my screen. This. All right. This is a US ABO question. Question 61. I forget which test, but one of the
Wait, how
tests.
many questions are there? Isn't there 50?
For the local round, there is 50. For the open round, I should say. For the semifinal round, which this is from, there is usually approximately 120 questions. And then there's also that's in the multiple choice. Then there's also going to be some kind of additional section. For example, this year, the additional section was a few of true false things. And then there was a few more matching. And then there was a few more heredity, open-ended short answer things. So just a lot of stuff at the end of these 120 questions. 120 is the average for multiple choice. There could be more. Yes, there is a jump from 50 to 120, but the 50 question part is with 50 minutes. The 120 is, I think, with 3 hours, if I'm not mistaken. So you do have more time. Anyway, it's 120 multiple choice with also, this is not your regular multiple choice. This is like you can choose multiple answers and then you have to get all of them right, so on and so forth. Not the easy kind, you know. And then there's also usually for after the 120, there's also the open-ended. Usually it's open-ended. Anyway, that's US able structure. So with this particular question, not fully doable yet because we didn't talk about any of these antiporters or ATP ACE pump. Actually, this one we should be able to do. ATP ACE pump. Thinking about what we talked about today, what do you guys think that is? First, there's the name pump. Then there's ATP.
Wait, doesn't that just like isn't? Oh wait, no, I think I'm getting this mixed up with ATP synthase.
Yeah, maybe. Don't overthink this. First, pump. What is that? What do you associate with pump?
Active transport.
Active transport, yes. ATP, that's something that we do need in active transport. When it says ATP ACE, ACE, you guys might know as like lactase or
Enzymes.
enzymes, yes. This is an
Breaks
enzyme.
down ATP.
Yes, breaks down ATP. ACE generally means an enzyme that does break down stuff. Not always, but for majority of examples I can think of right now, ACE, if something has the word ACE under it, it breaks down something. Lactase breaks down lactose, for example. So here we have ATP being broken down. And this actually makes sense if you think about it, right? When you need energy, you break down ATP, right? The ATP molecule has energy in it. Breaking it apart releases that energy. So ATPase basically means anything that consumes energy that is a protein, right? You consume energy, ATPase, you break apart ATP to get that energy. And this pump will need energy from ATP. It breaks down ATP to produce energy. Yes, active transport, ATP energy, yes. So Na plus K plus ATPase pump is an active transport pump. Now, this particular pump is a very famous one where there was like some ratio. I think it was like 3 Na plus to 2 K plus. Basically, both of them are moving in opposite directions so that you have a concentration of Na plus, a very high one on one side, and then a very high concentration of K plus on the other side. But since you're moving 3 molecules here and 2 molecules here, the net change in charge is happening, right? So that's why you're able to have a membrane potential. And this is very important for neurons, which rely on membrane potential to propagate signals. It's also important for many cellular processes like the release of Ca2 plus. I think that's what's going on here. The release of Ca2 plus, because heart cells also send signals, right? Your heart beats with synchronization. Your heart cells have to stay together and all that. So Ca2 plus is very active in that process. So yeah, basically this question is asking about what's going on. Since we're kind of running out of time, I'll just touch on one last note. If we're looking at this question, which I hope you guys have read by now, I know I was talking a lot, which might have made it hard, but if you guys have read this question by now, you'll know that it's about a heart cell that has this pump, this antiporter. If digitoxin, an inhibitor of this pump, is administered to a patient experiencing cardiac arrest, what will result? All right, so here I just want to demonstrate one thing about reading these answers. Muscle contraction is sustained. Muscle contraction stops, and it stops. And this one's kind of like your none of the above answer. It's like, oh, it will only affect this. None of the above answer. 9 times out of 10, your none of the above answer is not going to be the correct answer with any kind of USA book question.
Shouldn't it be ceases?
There are very rare cases where it is, but it's not. You're saying that it ceases. Well, if you have cardiac arrest, and you administer a certain kind of medicine, would you want your muscle contraction to cease?
Oh no, never mind. No, that doesn't make any sense.
All right. That's 2 answers, people that have put cease. One quick moment. I'm gonna check the answer key because I'm not gonna be able to solve this question this fast. No, it is not C. So for those of you guys who have said C, I'm sorry. It is B actually. So good job, Larry. I'm not sure how you solved that, but very good job nonetheless. What I was gonna point out is that this patient experiences cardiac arrest and you're giving a certain thing to it. If his muscle contraction ceases, then that patient dies. It doesn't make sense for this particular question. And that's why if I were doing this question, I'd immediately only consider A and B. Now, I wouldn't entirely rule out C, D, E. If A and B both don't work, of course, I'm gonna look at C, D, E. But just in the 5 seconds I would take to read this question, I'm already tending towards A or B, one of them.
How do you tell
So-
which one is right in between those?
Then we'd have to start talking about how to solve this question. Do you guys want to stick around for that? Because I know it's already 2 minutes
Yes.
after. All right, then I'll quickly go over this. An antiporter basically means that these two are moving in different directions. So that's what we see right here, right? Ca2 plus is out of the cell. Na plus, which you have 3 of, it is moved into the cell. Now with Na plus K plus ATPase pump, what usually goes on is 3 Na plus, I'll just put N for that, 3 Na plus move out, 2 K plus move in. And this is something that needs energy. This is not favorable. And that's how you build up a lot of Na outside the cell and a lot of K inside the cell. And that's why they're utilized for other processes. So you'll often see that if you need to move something else that's against this concentration gradient, you might just move some Na plus into the cell, and therefore you'll have the energy to do what otherwise would not happen because it requires energy. Similar thing with K plus, you usually just move it out of the cell, and then you get energy in that process. Therefore, you can do some other energy intensive process. So by having a lot of K plus here and having a lot of Na plus here, you can do all these processes. I briefly mentioned that this is very important with neurons. That this is how neurons happen. Na plus channels, going back to channels, simple diffusion, channels will open up, all the Na plus outside, which there's a lot of, all zooms back in, therefore polarizing the cell because now it's got a lot of positive charge, right? That signal, the positive charge is actually what travels along a neuron. That's what the signal is propagating. And then you restore that signal by then shooting in K plus to balance it out. Sorry, not shooting in, sorry, shooting out K plus, my bad. Shooting out K plus, charge balances back a little bit. Then you equilibrate the concentrations, pump reactivates, and then you form a depolarized state again. That's neurons. Anyway, back to this specific question, we can see that they're utilizing the fact that there's a lot of Na plus outside the cell to their advantage in order to put out Ca2 plus. So digitoxin inhibits this particular process, right? So you're not going to be putting out as much Na plus if this pump stops working. Therefore, cytosolic Na plus raised. What cytosolic means means inside the cell because the cell has the cytosol, right? So inside the cell, this very blocky cell. This whole thing is called cytosol, right? All the liquid inside. So you will now have more Na plus in the cytosol because you're not pumping it out, right? So that's why the cytosolic Na plus is raised and muscle contraction is sustained. Make sense?
Yes.
All right. That was a very quick summary of that based on the principles, by the way, that we talked about today. Now, I don't know anything really about the Na plus Ca2 plus antiporter. Looking at this question, I wouldn't have known anything about it, but through what I know about pumps, passive active transport, I can figure it out. So yeah, some applicable abilities here, unlike if I showed you a structure of the brain and then told you to just look at it and memorize its parts. Anyway, that's why I'm a Ken guy. That wraps us up for today. And I hope that was helpful. Passive active transport, phospholipid bilayers. These are the main topics we talked about today. I want to ask you guys now, would you guys prefer to talk about chemistry things or biology things for next week? What do you guys prefer? Chem? All right. All right, so I've got quite split answers. In which case, I'll see. I'll see what we work on. Because I did compile some other things, some other bio topics that we could have gone over today. Of course, I wasn't sure about how timing would work. So it looks like I over prepared. We can go over those or we can do some chem. I'll see what we do next week. Anyway, that's all for this week. And thank you guys for coming. And hopefully I'll see you guys next week. All right. Bye-bye.
Bye. Thank you.
Yeah. No problem, guys.